I don't have anything to be disclosed. I'm very happy here, and very exciting stage, and very exciting session of the SMSNA. I'd like to talk about how the parasites in erectile tissue was identified and characterized. Usually, majority of those from the data from my lab. As you know, until 2 decades ago, the endothelial dysfunction was regarded as mainly responsible for higher prevalence of ED, which precedes other vascular events of the human body. Those days, fortunately, we had some important researches from parasites of the human vascular system. For example, the parasites have a critical role in cardiovascular development and homeostasis, and those are required for tight junction integrity of blood-brain barrier, and critical for the regulation of vascular stability, and also regarded as a potential reservoir of mesenchymal stem cells or progenitor cells. With these knowledges, we have hypothesized that cavernous sinusoid has a compromised or poor barrier function that blocks the anterior blood component into the wall of the sinusoid. So our experiment shows that, the mouse experiment showed that endothelial cell-to-cell junction proteins were markedly sparsely distributed, and compared with those of blood vessels in healthy mouse. Moreover, in diabetic condition, the attenuation of the cell-to-cell junction was greater in cavernous sinusoid than those of other blood vessels. So, moreover, in disease with diabetes or dyslipidemia, the increase in vascular permeability to fluorescent tracer intravenously infused or to oxidative LDL in the cavernous sinusoid was found, while no significant changes in heart and limb tissues were found. This is, fortunately, we have identified and confirmed the presence of parasites in the penis of a normal mouse. You see the Ng2 positive red one, Ng2 positive parasites are densely and abundantly expressed in subtunical area. Usually there are microvessels, and then dorsal nerve and dorsal vein, and you see the inside of the corpora, the central part of the cavernous tissue shows less abundant, less density in parasites. We further localized the parasites from the human corpus cavernosum. You see here this in highly magnified field. This is a single sinusoid. You see the Ng2 positive parasites wraps around the abluminal part of the endothelial cells. And we further isolated and cultured the parasites from mouse and human corpus cavernosum successively. Primary culture parasite passage 1 shows a typical multidirectional projection of parasites, and is highly positive staining for marker NG2 and PDGF receptor beta, but did not show any positive staining for an endothelial cell marker. Primary culture, the pericytes maintain their cellular characteristics up to passage 2 or 3. So let me continue my talk to show the role of the pericytes in the pathophysiology of erectile function and dysfunction, and with its clinical implication. As you see, the pericytes expression is downregulated in both culture cells and the corpus cavernosum of diabetic mouse. So we have studied the role of pericytes in penile erection. Looking at the loss of function study, APB5 is an anti-PGF receptor beta blocking antibody was found which is intracavernously injected. It reduced the pericyte expression and penile erection in normal mice. So that means pericytes has positive role in penile erection. What about the gain of function study for pericyte? Fortunately, we had a powerful recombinant angiopoietin 1. This was developed by Go. This is known as an anti-permeability factor and mainly expressed in both human and cavernous pericytes. And so if it is intracavernously injected, it restores the tight and adherent junction proteins, and which decreases the extravasation of rhodamine oxide or oxide LDL level, and finally it improves the erectile function in ED with diabetes. What about if we inject the pericyte components? For example, pericyte-derived DCOF2 protein. It is also highly expressed in pericytes, and it was found to mediate the interaction between pericytes and endothelial generation, usually as shown in the left side panel, usually signal from pericyte to endothelium. And even enhances neurotrophic signals and promote to regenerate damaged neurovascular erectile tissue in a diabetic mouse, even in cavernous nerve injury model. But we don't know how it is communicated between the pericyte and the nervous cell and so forth. So we have extracted the pericyte contents using the pericyte-derived extracellular vesicle or mimetic nanovesicles. If it is injected intracavernously, it was also found to in the mediation of the LCN2 pathway, and finally to promote cell survival and the regeneration and suppressing the cell death signals. So it was found to promote neurovascular regeneration in the recovery of erectile function in a mouse diabetic model. Recently, we have an idea to the heme-binding protein 1. It is poorly understood in the peripheral nervous system, usually studied in CNS. To test whether this kind of the protein from pericytes has such a positive function, we injected it into the corpus cavernosum. And so the parasite-derived extracellular vesicles, including heme-binding protein 1, was found to improve erectile function in cavernous nerve injury-induced model. Therefore, we think that extracellular vesicles or endogenous protein from parasite can be a therapeutic target for ED in near future. So recently, Tom Lu group and our group have studied in single-cell transcriptome analysis from mouse tissue. Dr. Lu group, the human tissue. Our group, mouse tissue. So you identify the cluster of parasites from 15 cell clusters, and based on transcriptome patterns, and cell types were annotated based on the expression of marker genes. We can find the cluster of parasites here. And the gene sets related to vascular and leukocyte regulation were enriched in parasites. And the heart gene is more exclusively expressed in parasites. So this LBH we thought is also can be regarded as a marker of parasite in the human corpus cavernosum. As you see here, LBH is excessively, exclusively expressed in parasite rather than smooth muscle or other cells. So you see here as a from the mouse penis. So this was also found to increase parasite expression and penile erection in diabetic mice if it is injected intravenously. So this is the summary, how the parasite has a role in penile erection. The parasites are differentially distributed in penis, and the role of parasites of great importance in cavernous hemostasis and pathophysiology of penile erection and dysfunction. Especially these cells regulate endothelial cell permeability by controlling endothelial cell junction. And these communicate with endothelial cells or other cells to regenerate neurovascular structures, which remains to be elucidated further. Manipulation of the parasite function can be a therapeutic option, and we need a better understanding of a cellular relationship within the penis with parasite and other cells, and for further clinical application of targeting parasites. Our lab members, this person is usually involved in the parasite researches. Thank you very much.